Okay, welcome back. This will be video number 3 for the AEMT lecture series. Video number 3, I think in video number 2 we had just left off with the talking about the electron transport chain, talking about glycolysis, etc. So we're going to pick up where we left off there. Sorry about that. We had a problem when we were videoing, so we just roll with it. But yeah, we're going to pick up right there. We're going to start with cellular transport mechanisms. Okay, so let's talk about it. There's got to be a way for us to move things inside and outside of the cell, and there's a few different ways to accomplish that, and that's what we're going to talk about. Okay. So let's talk about the cell membrane. It separates the chemical reactions occurring from inside the cell from the chemicals that are floating on the outside of the cell. So it protects the cell, right? We know that the fluid inside the cell is called the cytoplasm. The membrane holding the fluid in the cell is, of course, the plasma membrane. All right, and we know that surrounding that is the matrix. The cell membrane helps to maintain homeostasis, and then we need to remember the definition for that. You may see that again. It's called a homeostasis, a normally balancing condition of the body that is resistant to change. All right, we have electrolytes. They're chemicals that are dissolved in the blood and are made up of salt or acid substances. Okay. So yeah, that's why they call this membrane a selective membrane, because it's very selective what it lets in and what it lets out. Okay. So diffusion. Okay. Sorry, hang on one second. I'm just looking at some notes real quick. So when we talk about diffusion, what we're talking about is sometimes the organisms, they need to be able to move molecules from the area of higher concentration to an area of lower concentration so that these solutions can spread out. And we talked about what diffusion means for the lungs, right? I mean, we talk about the concentration of O2 going in from the alveoli to the cell. So it comes into the cell because it's moving from a higher concentration to a lower concentration. So that concentration of oxygen is spreading out into the cell. And the same for the CO2. CO2 is going to move into the alveoli because it's moving in from a higher concentration to a lower concentration. Okay, it needs to be able to spread out. Diffusion doesn't cost anything. Okay, it's free. It's moving down the gradient. It's just like the river flows. You know, it's going to flow because that's what it does, and there's no energy cost there. All right, so when we talk about filtration, what we're talking about is the membrane of the capillaries is usually very thin. Okay, it's very, very thin, and so things can diffuse across that membrane as long as they're small and if they have some velocity to it, right? So an example of that would be like your kidneys, right? So you have the velocity of fluid coming through. It's small solutes, so it's just going to be filtered from that point on. Now it can't be large particles, because then it will not go through the membrane itself, because the membrane acts like a filter paper, kind of like it allows fluid to surround the body cells and keeps large molecules from getting into that tissue fluid. And so, just like I said, the kidneys filter compounds from the blood. Facilitated diffusion. So again, this is going to be free. There's no energy cost, but there's some other players involved, because like I said earlier, if those molecules are large, it's not going to cross the membrane by itself. It's going to require a carrier molecule to be able to move that substance in or out of the cell, from areas of higher concentration to an area of lower concentration. Now, when we talk about active transport, that's a different story, because active transport it's gonna require some energy. Okay, active transport is the movement of a substance against the concentration gradient. So it's like the river flows perfect as long as it's going in its normal direction, but we can't turn the river around to get it to flow the other way. If we do, there's going to be a cost for that. We're going to have to do something that's going to require energy. We're going to have to go inside, dig a dam. We're going to have to do something that's going to cost some energy. So then we talk about excuse me, then we talk about endocytosis. All right, and there's three forms of endocytosis. There's pinocytosis, phagocytosis, and receptor-mediated endocytosis. And that's just forms of active transport of how these things get across the cell. I'm sorry, how they get across the membrane. So here's an example of facilitated diffusion. We're just going from an area of higher concentration to an area of lower concentration. And then we can compare that with active transport. So we have something here outside the cell. It's going to come in and it just goes straight through, no problem. Didn't cost us anything. Now here, this iodine ion, it's going to have to be transported by a receptor site, and the receptor site recognizes this iodine ion and will allow it to come in, and active transport would mean that it would have to actually surround the ion once it landed and was recognized, and then transported from there to inside. And you'll see that the energy requirement here, you see nothing here, but you see your ATP. Here's your energy, here's your phi. That's your ATP kicking in and allowing that to pass through. In facilitated diffusion, we have a site here, a binding site. The solute comes in and binds, but is allowed to pass through freely with no cost. Osmosis. So when we're dealing with osmosis, there's a couple of things that we're dealing with that makes it different from diffusion. One is water. Okay, so usually the solute needs to be water. And the other thing is there needs to be a membrane involved. So there's your membrane right there. So that's when we're dealing with osmosis, that's how it differs with diffusion. Osmosis deals with water and membranes. And the reason why that is because there's different percentages of water when we talk about sodium levels. We know that normal saline is if I'm not mistaken, 0.09% sodium. So that's equivalent to cellular fluid. But if we go higher or lower with our sodium levels, then we're going to have to deal with what it's going to do to the cell. So here we go. So when we talk about osmosis and we talk about tonicity, so that's the concentration of a solution or its ability to draw or give water. And so what we look at here, we know that the blood in the body contains a certain amount of salt and sodium, salt, and the normal concentration is called isotonic. So isotonic is right here. That's the normal cell. All right, so if the concentration of the salt rises, the bloodstream becomes hypertonic. So here we have hypertonic. We have an increase in amount of salt. Okay. And then what happens is that excess of salt will force water out of the blood cell. Okay, so we're forcing water out of that blood cell in attempts to maintain an isotonic state. All right, so if too much fluid in the bloodstream, the blood cells have few molecules of salt in comparison. So over here, you'll see this one swollen. We have too much fluid. And we have an excess of fluid. We don't have a lot of salt, a lot of sodium. So the water is going to stay right here because water follows sodium. If there's no sodium, it's going to allow water to come in. So then the blood cell attempts to take in water and attempt to normalize the blood to make it isotonic, but doesn't work out too well. And then what ends up happening is the cell will rupture or burst, and that is known as lysis. Lysis is too much water entering a cell, and then it will swell and burst. I would remember that term, lysis. If too much water enters a cell, then it will swell and burst. So intracellular fluid, the body's fluid is divided into two main components. We have intracellular fluid and extracellular fluid. Remember this, interstitial fluid is located in the spaces between the body cells in the extracellular space. You'll see that again. So interstitial fluid is located in the spaces between the body cells. That's known as the extracellular space. Alrighty, so there's, hang on, just let me make sure. I'm going to kind of cruise through here because we've kind of already covered a lot of this, but I will let you know. So the function of the skeletal system, all right, gives us our recognizable human form and protects the vital organs. Okay, remember that. Remember that. All right, the axial skeleton. The axial skeleton is composed of the skull, thoracic cage, and the vertebral column. I don't want to keep hollering Behoover, so, but everything that I'm going to say, if I'm kind of cruising through something like this and say, well, wait a minute, let me look, you can bet you'll see it again. So I'll say it again. Axial skeleton is composed of the skull, thoracic cage, and the vertebral column. All right, so we should know this already, but the brain and spinal cord are connected through a large opening at the base of the skull called the foramen magnum. Okay, remember that. Remember what the foramen magnum is, and that's that large opening at the base of the skull. Remember the cribriform plate. Okay, so what is the cribriform plate? It's horizontal bone through which the olfactory nerves filaments pass.